Alright guys, hey, I'm going to introduce some of the models and terminology we're going to look at in lab for the respiratory system. We'll start with the sagittal head section. On this section you can identify several things. Let me get my pointer here. The external nares are the cartilaginous openings that go into the nose that let air up into this waiting area called the vestibule. The vestibule leads into the nasal turbinate, which consists of the three bony concha, superior, middle, and inferior, and then the grooves that pass between them called the meatuses. When we go posterior to the turbinate, we get into the choana, and the choana contains the opening of the eustachian tube. And then we pass into the nasopharynx, the muscular tube here. We can see the smooth muscle on the walls of it that contain or house the adenoids, the pharyngeal tonsils, and then the uvula of the soft palate. And that whole area there is the nasopharynx. And then we come down into the oropharynx. And there we have the palatine tonsils and the lingual tonsils. And then we come down a little bit below that. My picture got a little cut off. But we come down below that and we have the laryngopharynx, which contains the opening into the larynx anteriorly. That opening is called the glottis, and therefore the cartilaginous flap above it is the epiglottis, which you can see here. So we just start to see the tip of the larynx on this model. And then the whole model extends down. You can see the entire larynx. And I wanted to point out, when I posted my picture in here, it cut the bottom of it off. But if you come just a little bit down into the larynx, you'll see two little like puckered lips here. And the upper one is the vestibular fold, and the lower one is the true vocal fold. The vestibular fold is a fleshy fold that collapses into the middle of the larynx when you swallow, and the epiglottis folds down and lays like a lid over the vestibular fold, and those two seal off the opening of the glottis so that foodstuffs being swallowed pass over that opening to the larynx and into the esophagus. It keeps you from aspirating material down into the lungs. The lower of those two folds is the true vocal fold, and that's actually what vibrates. That's a ligamentous structure that vibrates as air is passed over it, and that creates the different tones of our voice. And we have a trachea model I'll show you in a sec. Here's just another view of the nasal and oral cavities. This is on the torso model. So again, we can see the concha and the meatus here and the external nares. This would be the choana. Then as we pass into the oropharynx, we have the pharyngeal muscles intact on this model, so we can't see the oropharynx because it would be deep to this. But as we come down here, this is the area where we can see the larynx. And that's the thyroid cartilage right there. And you can take this off and look inside, and that's what the next picture I have is. With that, now that the lateral wall of the larynx has been removed to show us the thyroid cartilage anteriorly. And this is the thyroid prominence that's tipping forward here. That's the Adam's apple that is more protruding in males than in females. And that sits on a cartilage here that it wraps around the base, which is called the cricoid cartilage. These are the folds I was talking about that you can see on the other model as well, the sagittal head section. This top one here is the vestibular fold, 208, and 209 would be the vocal fold. And that again is your voice box or your larynx. And that sits atop the trachea. So these white structures here are called annular or C-ring cartilages of the trachea. They go roughly 3/4 of the way around, but then they remain open in the back where there's a muscular wall called the trachealis muscle. And here we move down the torso a little bit to see the lungs. And so we're into the thoracic cavity, and here we can see the ribs cut through with their intercostal muscles in between, with an the inner layer of the internal intercostal muscles and the outer layer here of the external intercostal muscles between the ribs. We see the lungs themselves with their broad base and their cone-shaped apex. The base sits on the diaphragm. The apex is up under the collarbone. The lungs happen to fill from the base towards the apex. And your right lung is a little bit larger than the left lung. The right lung consisting of 3 lobes, and here we can see the prominent superior lobe, separated from the middle lobe by what's called a horizontal fissure. And then this is the middle lobe right here, separated from the inferior lobe by an oblique fissure, which is more lateral, and it wraps around the back, so we can only see the front part of the oblique fissure, but if you were to look at the side of a lung, this wraps all the way around the back. So actually the inferior lobe is one of the larger lobes of the lung. So the right lung has 3 lobes. Left lung has 2, a superior and an inferior, separated from each other by an oblique fissure on the left side. The left lung, you might notice, has an indentation here called the cardiac notch, and that's where the apex of the heart is tilted that direction. The indentation on the medial aspect of both lungs is called the hilum or hilus, and that's an indentation inside, which you really can't see from this point of view, but it's in there. And that's where the pulmonary arteries enter, pulmonary veins exit, and the bronchial tree enters into the lungs at the hilum. And the lungs, at their base, they sit on the diaphragm. Here we've taken the anterior wall of the lungs away to show us what's underneath, and most notably we can see these ribbed structures back there underneath the vessels, and those are some of the bronchial trees. Those are actually bronchi, tertiary bronchi. The blue and the red, well, the blue represents the pulmonary arteries and the red represents the pulmonary veins. Of course, the heart sitting prominently here in the mediastinum within its pericardial cavity. So now we've taken the heart out, so we can visualize what's behind it. And right here, we just catch the bottom of the trachea. Descending down, the larynx would be up here, and we come down, and there's the trachea. This large cartilage of the trachea at the base of the trachea is called the carina. It's shaped a little bit like a diaper. That shows up on an X-ray radiogram that's radio-opaque. So that's a landmark that they can usually tell where the bronchial tree enters into the lungs when they can see the carina. This is a right primary bronchus on this side, and the left primary bronchus over here. And you can see that they're banded by full rings of cartilage that go around. And then we get to this level, and they start to bifurcate. The right lung has 3 lobes, so there are 3 lobar or secondary bronchi, 1 and then 2 here, and then the third one goes down there. And then when they split again to about 8 different segmental or tertiary bronchi, and that's what these little guys down here are. On the left side, the left primary bronchus actually curves and extends a little bit farther than does the right. The right's a little wider and more in line with the trachea. So if things are aspirated down to the trachea, they most often lodge in the right primary bronchus and not the left, because the left curves more. The left lung has 2 lobes, and so we really can't see the lobar bronchi, but they'd be here. There'd be one there, and then they go off, and then what they've shown here are tertiary. And they're showing you the branches again in blue of the pulmonary arteries and the red the pulmonary veins. The base of the lung sits on this fibromuscular structure here, the diaphragm. And just to reiterate again, so you get this, when the diaphragm is relaxed due to elastic recoil of the lungs, it's dome-shaped. Okay, it's up like this when it's relaxed. When it contracts, it flattens down and it drops the floor of the thoracic cavity down, effectively expanding the thorax. That's when it contracts. When it relaxes, it goes back up. Okay, as promised, here's our trachea model. Here's an anterior view. We're looking at the hyoid bone. If you remember, that is the bone that's just really pretty much at the base of the oral cavity and helps root the tongue in place and helps hold some of the pharyngeal muscles that are involved in swallowing. So that's the hyoid bone. That's connected via a membrane down here to some of the cartilages of the larynx. So the most prominent cartilage in the front of the larynx is the thyroid cartilage, this butterfly-shaped thing here, which has a thyroid prominence or an anterior prominence that sticks out the front. And that's what tips forward in males as they go through puberty and accounts for the deeper voice that males have in the presence of what we would call the Adam's apple. The base of the larynx is the cricoid cartilage that we can see right down here that wraps all the way around. And there's a ligamentous membrane that connects the thyroid cartilage to the cricoid cartilage, and that's called the cricothyroid ligament. And that's worth mentioning because if somebody has to have an operation to bypass the larynx, whether they're having some sort of tracheal cancer or laryngeal cancer, or just bleeding or something that they have to bypass the larynx for, or maybe even an occluded airway, you can puncture a hole in that cricothyroid ligament and insert a cannula, and that's called a cricothyroidectomy, and usually just called a cric tube. And that would allow somebody to breathe, bypassing the nasal passages and the larynx. Beneath the cricoid cartilage, we get into what are called the annular cartilages of the trachea. There are roughly 20 of those as they extend down to the carina where the bronchi will be. And here we see the thyroid gland just a little bit inferior to the thyroid cartilage, having been cut through. Sticking up here at the top like a little tongue, this is actually the epiglottis. I'll have a better view of that in this picture. So here we're looking from kind of a superiolateral point of view. There's the epiglottis, hyoid bone. So when you swallow, the epiglottis folds back here and covers the opening of the larynx. What we see in the back, well, here's the trachealis muscle that covers the posterior of the trachea. Those are the C-ring cartilages that come around till they meet the trachealis muscle. You can see the thyroid gland wrapping around here. The base of the larynx is the cricoid cartilage, and see how broad the cricoid cartilage is in the posterior. It gets quite narrow in the front, but it's very broad in the back. The thyroid cartilage slips over the anterior aspect of the cricoid cartilage, and that actually slides up and down when you swallow. Here are two more cartilages you should be familiar with. There's a dividing line right about there, and this is just one tiny little beak-shaped cartilage. That's called the corniculate cartilage right there. And then the one that looks like Olaf the snowman right here, I know that's just in my imagination, but that one right there, that's called the arytenoid cartilage. Well, the arytenoid and the corniculate cartilages are connected to a series of muscles which make them pivot back and forth, and you can kind of work that in this model. You can see how these swivel this way. Well, when they pivot back and forth, they change the tension on this white membranous structure here, which is supposed to represent the true vocal cord. So when you stretch that, that changes the pitches of the voice. A little bit tighter stretch and you get higher pitch, and then you relax it, you get a lower pitch. And the length and thickness of it determines the depth of the voice. Above this, in this area right here, see the shallow fossa right there? There's actually a fleshy membrane that's attached there, and that's the vestibular fold. It's not pictured in this model, but it would be right there. When you swallow, these two vestibular folds, they collapse into the middle, and they almost form like lips, and then the epiglottis lays down like a lid on top of that, and pretty much effectively seals the opening of the larynx, so that material being eaten or drank will pass over that and then go into the esophagus posteriorly. Interestingly, so the esophagus is right up against this trachealis muscle in the back, and if you've ever taken a larger bite than you intended, or a larger swallow of something than you intended, and it hurt like crazy as you swallowed it down, what happened there was that bolus of food passed down the esophagus and stretched these C-ring cartilages, and that was the pain that you felt, was the distension of these cartilages being moved out of position by the bolus of food passing posteriorly. So that really, really hurts. You can imagine why one of the self-defense techniques that is frequently taught is to just hit somebody with the side of your hand very hard right beneath the Adam's apple and hit them in the trachea, and that's excruciatingly painful. Here's a superior view, just looking down so we can more clearly see the true vocal cords. So just to make sure you understand, what's shown in this model is supposed to be the true vocal cords. The vestibular folds would be above this, superior to that, and they're not shown. You can imagine where they would be right here, but they're not shown. There's one more cartilage of the larynx, in addition to the cricoid, thyroid, epiglottis, arytenoid, and corniculate. It's called the cuneiform cartilage, and it's a lateral cartilage that runs in this area, and I've oftentimes wondered in this model, so I don't really have a key for it, if that's what they're trying to indicate with this little white blob, even though they've painted that like it's a bone. Because this is the right area for the cuneiform cartilage, but I never asked that on the test because I'm really not sure if that's what they're trying to show or not. Okay, here's our lung board. So the lung board is a good place again to see the larynx. That's a miniature version of the larynx right there. We're not going to go over all those structures again. You can see the larynx sitting on top of the trachea and the annular or C-ring cartilage that descend down here. We can see our right lung with its superior, middle, and inferior lobes, left lung with our superior and inferior lobes, and the fissures, cardiac notch, diaphragm. Here we've taken away the anterior aspect, and again we're seeing the divided lobes. This one is going to show us on the next slide a little bit better representation of the bronchial tree. As we come down the trachea, there's that diaper-shaped cartilage called the carina. You can see the right primary bronchus, kind of wide and straight. The left one's more twisted, more curved. We get to these initial branches on the left. Those are the lobar or secondary bronchi. And here are the secondary bronchi on the right, 1, 2, and 3. And then when we get up to this area, these are the tertiary bronchi, called segmental bronchi, of which there are about 8. Well, let me back up here. So we go to the tertiary bronchi, and then we start branching into a system of things called bronchioles. And bronchioles are just any air passage that's smaller than 1 millimeter in diameter. And there are 23 levels of branching from the primary bronchus all the way down to the last one, which is called a terminal bronchiole. And so those are called generations of branching or levels. So here we go. There's 1, 2, 3. Then we go another 20 bifurcations or splits. And we get smaller and smaller and smaller to the point where we have a lumen size that's only a matter of, say, 10 to 30 micrometers in some of the smallest bronchioles. So that's what they're trying to indicate here. This structure up here with the blue colored cartilage plates would be pretty much the last branching of the bronchial network before we get to what's called the terminal bronchioles. So right here, sticking off the end, that would be a terminal bronchiole. This would be a terminal bronchiole. We know it's a terminal bronchiole because there's still cartilage plates associated with it, which you'll still have present with the terminal bronchioles, but you will not have present with what are called respiratory bronchioles. The kind of orangish pink bands that run around this, that's supposed to represent smooth muscle. So on this model, I always tell students that to consider the ones that are cut off that have the blue around it still to be terminal bronchioles. And that just means that's the last branch of the bronchial network. Some other things that are present here, well, we can see the blue blood vessel coming in, that's the pulmonary artery, and the red blood vessel going out, that's the pulmonary vein. Down here shown, these are pulmonary capillaries, and they're just trying to show pulmonary capillaries in their deoxygenated form and in their oxygenated form here. Wrapping around all of these things that look like tiny little mini marshmallows, well, the mini marshmallows are air sacs. Those are called alveoli, a singular alveolus, of which that makes up most of the tissue of the lung. Down here, shown with these little blue dots, that's actually the outer membrane surrounding the lungs. That would be the visceral pleural membrane. And you go beneath that, and we get the black fiber there. That's elastic fiber, the external elastic membrane. And then we get collagen fiber in the fibrous membrane. And then we get some more elastic fiber in the internal elastic membrane. And then those are the outer layers of the lung. If we rotate the model around a little bit, we'll see this interesting spot here where we've cut open the ends of some of these alveoli. When we have a tight cluster of alveoli like this, we call that an alveolar sac, and so that's what we're outlining right here. Just cut through that sac, and you'll see there's this darkened tunnel in the middle of the sac. That's the end of what we call a respiratory bronchiole, and I'll explain what that is in just a minute. Oh, actually, I'm sorry, that's not the end of a respiratory bronchiole. That's the end of an alveolar duct, which comes from a respiratory bronchiole. And I'll show you a blow-up of this alveolar sac in a minute. We'll talk about the cells that make up the walls of the alveoli. Now here I've rotated the model around to the back side so we can look at it. And again, nothing new. We've got the bronchial network up here, the terminal bronchioles there and there. And then what we have right here cut off and extending down here, these are respiratory bronchioles. Now we know they're respiratory bronchioles because A, there's no cartilage left associated with them, B, they have just smooth muscle, and C, and most importantly, they start to actually have some of these little alveoli attached. And I don't know if I can go back and show you. Yeah. So like right here, for instance, that's an alveolus that's attached to a bronchiole. That makes that a respiratory bronchiole. Physiologically speaking, this is an important demarcation because the respiratory bronchioles with the alveoli that are attached to them, that's the beginning of what we call the respiratory zone of the lungs. That's where we can start to have gas exchange occur. Prior to that, with the terminal bronchioles and everything proximal to that, that would all be the conducting zone and essentially makes up the anatomical dead space of the lung, where it's full of air, but there can be no gas exchange occurring. Okay, so we got the respiratory bronchioles here, and we start to have alveoli associated with them. And then you get into these areas where it looks like a long hallway. Now, disregard the pulmonary artery, which is the blue vessel here, but just look at the white that I'm tracing. See how it looks like a hallway? Okay. That's an alveolar duct. And the alveolar ducts come all the way down, and they terminate in a sac. So down here would be the sacs. All right, and before I go any farther with that, let's back up here and take a look at the blood vessels. Now you know this large blue one here is a branch of the pulmonary artery, and the red one over here is a branch of the pulmonary vein. So pulmonary artery is taking deoxygenated blood from the right side of the heart to the lungs to be oxygenated. It's oxygenated in the pulmonary capillaries and then goes off in the pulmonary vein, now oxygenated back to the left atrium of the heart, eventually for dissemination around the body. But take a look at these vessels that are drawn in light red and a lighter blue, kind of like an opaque red and blue. Well, those are bronchial arteries and bronchial veins. And perhaps you'll recall from lecture that the bronchial arteries are carrying oxygenated blood to the mesenchymal tissue of the lungs, in other words, the structural support tissues of the lung. And the bronchial veins are taking deoxygenated blood back from that same tissue. The bronchial vein happens to join with the pulmonary vein. And you might notice that there's an incompatibility there. The bronchial vein is deoxygenated blood, and the pulmonary vein is oxygenated. So that does have the effect of lowering the PO2, the partial pressure of oxygen in the pulmonary vein, just a little bit. I'll let you go back and review that in the lecture respiratory section if you want more explanation for the consequence or lack of consequence of that. So now I've spun the model back around and I'm looking at closer detail of that alveolar sac. So the alveolar duct that I just showed you on the back side, that ends here. And it opens up into this sac. Now the sac's been cut open to show us the walls. The red little dots are supposed to indicate pulmonary capillaries. And then all of the little yellow outlines in the back, they're outlining what we call squamous cells, type 1 pneumocytes. Type 1 pneumocytes are called the squamous pneumocytes, and they're the ones that do gas exchange. So this is the site of external respiration. Oxygen has to diffuse from the alveolar space through that cell and into these capillaries, and CO2 has to go from the capillaries through that cell into the alveolar space. That's external respiration, done by the type 1 pneumocytes. They're part of the respiratory membrane. Now the ones that are in pink here with little dots in them, those are supposed to be the type 2 pneumocytes, or what are called great alveolar pneumocytes. And their job is to secrete a proteinaceous compound that's called surfactant. And surfactant is basically a detergent that disturbs the surface tension of the water in the alveoli. It reduces the surface tension, so the alveoli are able to remain sprung open. They have a little bit of elastic fiber that surround these alveoli, and as long as the surface tension is reduced, the alveoli will stay a little springy and they'll stay open. Without the surfactant, the alveoli collapse, and they really just kind of take on the consistency of wet tissue paper. And if they collapse, then they're unable to do gas exchange, of course. All right, so your type 2 pneumocytes tend to be at the junctions where alveoli meet. And I always tell students they look a little bit like a robin's egg. They have a little kind of a speckled texture. They're a little bit larger and rounder than are the type 1 pneumocytes. And we'll have a chance here in a second to look at the histology slides of those. All right, into the histology. The first histology slide I want to remind you of is one we looked at in A&P1, when we looked at pseudostratified ciliated columnar epithelium and hyaline cartilage. So this is a trachea slide, and deep in the trachea, this is the layer of hyaline cartilage up here with its smooth, glossy hyaluronic acid and proteoglycan matrix, and the chondrocytes in their lacuna are the cellular aspect. We have a perichondrium that surrounds that, and then we get into this layer here, which is a submucosa. Embedded in the submucosa, we have many seromucosal glands, and that's what we're looking at right there and here and here. So that's what's manufacturing and secreting the copious amounts of mucus that we would find in the trachea. And then finally here, this is our pseudostratified ciliated columnar epithelial layer. You can just make out very closely the fine cilia along the edge there. Now we get into the lung tissue itself. I started with this slide because it shows some of the bronchioles. The white spaces here are alveoli. The small white spaces, like here and here and there, those are capillaries, but the larger white spaces are alveoli. And then these funky structures are bronchioles. You can see two in full view here, and down there's another one where we just see the cartilage plates around that. The dark blue staining fiber in here is elastic fiber. And then beneath that we have a little bit of smooth muscle. And then we have a sometimes columnar, sometimes cuboidal epithelium that actually lines the bronchiole itself. So this is a bronchiole. Now whether that's a terminal bronchiole or a respiratory bronchiole, it's a little hard to say. That's probably the level of something like a terminal bronchiole. In the respiratory bronchioles, the elastic fiber is usually not there. It's just smooth muscle. Down here we have a larger bronchiole, and we know that because that's one of the cartilage plates. So you don't have that below the level of a terminal bronchiole. So that's either a terminal bronchiole or something larger. Okay. And like I said, these are alveoli. Let's look at a closer picture of the alveoli. All right, this isn't the clearest picture in the world, but the white spaces are alveoli. Obviously, we've magnified the image a little bit larger. Right up here, where I have the pointer right now, or where I have the laser pointer, that's a type 1 pneumocyte. And here, this is more than likely a type 2 pneumocyte. I have a better picture of that later. Right, these are the walls of the alveoli, and they separate the alveolus from things like capillaries, which this is a blurry capillary right here. This is collagen fiber, so I think this slide is actually taken from fairly near the edge, or at least one of the septa of the lung, because that's collagen fiber. And then these are a mixture of fibroblasts, which are producing the collagen fiber and elastic fiber, capillaries mixed in there, you can't always make them out, and neutrophils and white blood cells are making up that mass of cells. Now here's a spot where we have a nicely isolated squamous cell. These are type 1 pneumocytes, very, very thin like that with a nucleus. There's a type 1 there, type 1, type 1, another type 1 down here. Right, and here are a couple more. So those are what make up this thin wall of the alveoli. Those are type 1 pneumocytes. Right. Type 2 pneumocytes you find clustered in areas like this. That's probably a type 2 pneumocyte right there. Again, that is too. I'll show you a better picture here. All right, now here I've just taken another slide. This is more than likely a type 1 pneumocyte up there. We can't really see any other ones. Some of these cells that line these are certainly type 1 pneumocytes, but you really can't be sure. That's probably one there. But you see this round cell? Okay. That's a type 2 pneumocyte. That's a great alveolar pneumocyte, as are those two. I picked this spot because there's a cluster of them out there on this kind of little projection. That speckled cell right there is a very nice isolated type 2 pneumocyte, no doubt about that one. Okay. See, they have a very different appearance of their cytoplasm than do the type 1 pneumocytes. Here are 3 of them clustered together. 1, 2, 3. Those are 3 type 2 pneumocytes right next to each other. So they're the ones that secrete surfactant. Oh, here's another alveolus. This would be a type 1 pneumocyte here. Type 1, type 1. Over here, right there, that for sure is a type 2 pneumocyte. That's probably a type 2 pneumocyte at the tip of the pointer. So type 2, type 2. There are also many monocytes, macrophages, that roam around in here. They're called dust cells, and I've never been terribly good at identifying them. That might be one there, that might be one there, that one might be one right there, but I say might be because I just have never been particularly good at actually identifying them. The function of the macrophages in the lungs is that they crawl around in the respiratory bronchioles, the alveolar ducts, and the alveoli themselves, and they engulf any foreign material that's gotten in there because there are no cilia below the level of the bronchioles. So if any dust or foreign material gets in there, the only way to get rid of it is for the macrophages to ingest it, and then they actually crawl back into the bronchial network where they get trapped in the cilia, and they are swept via the mucociliary escalator, which is just a bunch of cilia that sweeps all that up to the oropharynx, where then we swallow it down into our stomach and destroy it. Okay, that ends the show, guys. Hopefully you'll have a chance to work with the models and you can use this guide. I'll put the PowerPoint up there as well so you can look at the pictures without my narration. And if you have any questions, I'm sure Dr. Rex can point you in the right direction. Thank you so much.